prepare for the fungal member, the fungi, and another thing is the to develop to the this one. I'll share my PPT. Okay. Here, the fungal is how to use. These are things we will discuss. Today, we will discuss about to the what are the fungi, how to use. These are things, then antibiotics. The term is antibiotic, as it is origin and then the word antibiotics, that is the against life. Next one, the antibiotics are chemically substance obtained from the various species of microorganisms. Best example for the microorganisms: bacteria, fungi, actinomycetes. This is a best example for this one. That suppresses the growth of the organisms. Which organism? Microorganisms. And evenly may destroy them. The probably points of the difference among the antibiotics may be physical, chemical, pharmacological properties. These and every antibiotics, physical, physical one, chemical, and the pharmacological properties, antibacterial spectra, and mechanism of actions. They have made it possible to cure disease caused by bacteria, such as pneumonia, tuberculosis, and meningitis. They save the lives of millions of people around the world. What is the classification? The antibiotics are classified into main basic of their mechanism of action and by its chemical nature. The classification of based on the antibiotics. The first one is called the mechanism of action. Against that inhibit the synthesis of bacterial cell wall. These includes the penicillins and cephalosporins that are structurally similar and dissimilarly against, such as cycloserine, vaxomycin, bacterium, and imidalose antifungal agents. This is the mechanism one. Again, the second thing, against that, act directly on the cell membrane of the microorganism. In the microorganism, cell membrane, affectable and permeability, and leading to the strategy, leading to the linkage of intercellular components. These includes polymyxin, poly antifungal agents, niacin, and amphotericin B that bind is a cell wall sterols. Agents that life or that affect of the function of 30 to 50s ribosome submitted to subunits to cause reversible inhibition of the protein synthesis. inhibition of the protein synthesis. These includes tetracycline, erythromycin, chloromaphenol, slindamycin. These are coming the another thing. The agent that bind to 30S ribosome. Ribosomes is a two. One is the 30S and 40S. 30S and 40S, 50S and 60S. Ribosomal subunit and are point protein synthesis. These are the useful for the protein synthesis. Protein synthesis, these includes aminoglycosides. Aminoglycosides get aminoglycoside that leads to cells enter leads to cells enter leads to cells death evenly. The agents that affect nucleic acids metabolism. Here, nucleic acids meaning two types. One is the DNA, another one is the RNA. Both are found into the nucleic acid, but both are not mixed in the RNA polymers. The nucleic acids is that affect nucleic acid metabolism, such as ravifying, which includes DNA dependent RNA polymerase. The classification based on chemical structure. First one is the beta-lactam antibiotic. Second one, aminoglycan antibiotic, tetracycline antibiotic, macrolide antibiotic, other antibiotics. What are the beta-galactin? These consist of two major class of agents, that is penicillin and cephalosporesis. First coming to the penicillin. First one, penicillin, the most important antibiotic. It was the first identified or extracted from the mold penicillium notatum. Penicillium notatum. This is the penicillium is the most important. Penicillium, uh, one second. Penicillin is the most important antibiotic. Was first extracted from the mold Penicillium notatum. This Penicillium notatum, subsequently a mutant of a related mold. Another one is the Penicillium cryogenum, was found to give the highest yield of penicillin and is employed for the chemical production of antibiotics. This is the penicillium is a very important is that one belongs to group of the antibiotics called beta lactam antibiotics. The basic structure of the penicillium consists of a thiazolidine thiazolidine ring fused with the beta-lactam ring, which is essential for antibacterial activity. These are these two rings constitute the fundamental nucleus of the all penicilliums, namely 6-amino penicillinic acid, that is a 6-APA. A variety of semi-synthetic penicillins are produced by alternating the composition of the side chain attached 6-PAPA nucleus. Both the 6-APA nucleus and side chains are essential for the antibacterial chain. See, this one is the side chain and basic structure of the penicillin. Penicillins are named in the following ways. One is the chemical abstract. Second one, the chemical abstract is the first one. The penicillin are described as 4-thia, 1-azolo heptanase. Second one, benzyl penicillin is 6-2-penicillin phenyl lactomide. 3-dimethyl 1,7-oxo 4-thioazidine. In order to simplify, the unsubstituted bicycle ring system of penicillin. It is given name phenum. According the penicillins are 6-acylamino, 2-dimethyl penham 3 carboxylate. The classification of penicillin is suspectable penicillins. The general impact on antibacterial activity is as follows. This is the good gram positive potency against good gram against positive productivity positive gram against simplify are stably local and staphylococci. Next one, fourth useful against some gram positive cocci. Non-effective against gram negative bacilli. Second one, penicillinase resistant penicillin. General impact on antibacterial activity is as follows. Decreased susceptibility to many penicillin active against microorganism resistant to early penicillin. Oxalicinase offer good oral activity. Third one, ampicillin. Ampicillin is general impact on antibacterial activity is as follows. Extended spectrum of activity against some gram-negative bacteria and retention of gram-positive potentially. The infective against Pseudomonas aeruginosus. Coming to the next one, uridopenicillins. How to the penicillin work in the antibiotic. The mode of the action, the cell wall of bacteria is essential for the normal growth and development. Peptidoglycan is the cell wall. Peptidoglycan is a heteropolymeric component of the cell wall that provides ridge mechanism for sustainability by various various of is highly cross-linked lactis wise structure. The peptidoglycans is composed of glycan chains, which are linear strands of the alternation amino acids. This is N is equal N N-acetyl glucosamine and acetyl numeric acid that are cross-linked by peptidoglycan chain. Cross-linked by the peptidoglycan chain. Peptidoglycans chains of a enzyme transpeptide transpeptide. The penicillium inhibit the transpeptidase activity to the syllabus activity to the activity to the syllabi synthesis. The biosynthesis of the biosynthesis of the poly peptidoglycans the beta transpected enzyme that contains it is probably established by beta carotene enzyme improved that improve and my exhibit here the transcriptions the enzyme that contain protein synthesis that is transmitted my enzyme is that consists to protein is proteinized. The next one that's transmitted the clear NH3 NH3 and NH3 when the bonding and bonding ring. This reaction of the enzyme inconvenient are uh, the renders and inhibit of the polypeptide chain. Thus, the substitution of R on the primary amine with an electrons withdrawing group decreases the electron density on the side chain of the products. If you if you uh, RRA the substitution of R on the primary amine with an electron withdrawing group decreases the electron density. The electron density and interactive the electron ये हमारा इसमें मिरर वर्क के साथ जा रहा है the side by side mud the group of decreases the electron density on the side chain of protect them from acid degeneration. Substitute on the alpha region of the side chain. The maybe benzyl penicillin undergoes acid and make that is the one the here the thylakoid degeneration and susceptible for the no, no. 1/2, 1/2. Thus, in substitution of a bulk group on a alpha carbon of the side chain, side chain, side chain beta case around the egg. Hmm. The ring attached directly to the side chain amide carbonyl, and there is a substitution at both position, ortho, and point of the attachment. The size of the ring systems play an important role in the determining the ability of the ortho substituent to conform to Brazilium. Next one. These are the isomic form of the penicillin and the isometric form of a penicillin differs in their activity. For example, D isomer 28 time molecular that is the times more active than L isomer amoxicillin where the amoxicillin layer. The introduction of the polar group are recognized in the into the molecular ionized molecular the example D isomer is the 28 times more than active than L isomer of RT and molecule. In alpha position of the side chain, the beryl carbon is the penicillin G. The conforms against the gram-negative penicillin and hydroxyl, the conforms against the gram-negative uh, bacilli, amino hydroxyl, carboxyl, and sulfur, and carboxyl and sulfur. This one a replacement acyl side chain with a hydroxyl group shows improved gram-negative activity and num introduced 5,6 5,6 alpha metho group produce greater stability. The lactamase N-acetylate ampicillin, uridopenicillin have increased activity against Pseudomonas. Many enters of the carboxylic group pattern, the attachment of the C3 have been prepared and prepared have been prepared as a prodrug to increase lipophilicity and acid stability. For example, acetomethyl ester derivatives are used for preparing prodrugs. The sulfur anthozoid ring, which O plus CO2, which O for CO2 and H beta CH3 gives broad spectrum antibiotical activity. Hmm. The geminal dimethyl group C2 position is is a characteristic of the penicillin. This one is a characteristic of the penicillin. In general, in general, derived in general, derived in geminal dimethyl group at C2 position is a characteristic of the penicillin. And the in general cultivation, that is the general cultivation of the C3 carboxylide carboxylic acid functionally is not tolerant unless free penicillin. This is rapid cleavages in bio to generic activity of the penicillin. And for example, pavum penicillin and B-campicillin, the antibacterial activity is evident by N4 atom ring for the junction. In vitro condition, the degradation is related to retreated by the keeping the pH of the solution between 6.0 and 8.0. More lipophilic side chain increases. The plasma protein binding, for example, ampicillin, 25% plasma protein bound and phenoxymethyl penicillin, 75% protein bounded. And the next coming to the penicillin resistant and cephalosporin. These celebrations, the cephalosporin were isolated from the fungus Cephalosporium erysimonium in 1948. That is Newton and Abron in 1953. The main product being cephalosporin C, the molecular modification citation, and that is the ఇప్పుడు అన్ని కష్టపడాల్సి వస్తుంది. The penicillin, cephalus, the molecular modify action, that is the origin, they are beta lactam. And the main difference in between the two is that cephalosporin contain dihydromethagen. That is the important of that one. And the while penicillin contain a tetrahydroxylin ring. The cephalo and The cephalo, and that one is to repeated at the okay. Cephalo and penicillin have mechanisms, however, they bind in the target protein other than penicillin. The nomenclature of these are things. Penicillin are the nomenclature of this. Now, this is the total penicillin. Now we will discuss about to the industrial in fungal uses. How to use the fungal industrial. In the fungus, industrial methods are most important. That is fungal enzymes and metabolisms. And how to use the fungal metabolisms. These are things we will discuss now. Fungal metabolism and one second, I'll share PPT. Metabolism and fungal metabolism. Enzyme and metabolism. Fungal enzyme. Now I will share the one. One second. Here now chapter 1 to fungal physiology. Fungal physiology refers to the nutrition, metabolism, growth, reproduction, and death of the fungal cells. It also relates to interaction of fungi with their biotic and abiotic surroundings, including cellular response to environmental stress. Cellular response to the environmental stress. And next, which they are the industrial and process human health relation to the ecological accepts. The biogeological cyclings of carbon, oxygen, nitrogen, phosphorus. These are the cycles. These are all called biological and biological cycles. That one is the biological cycles. That all are the cycles for the biological and that one is the cycles. And death of the fungal cells, death of the fungal cells meaning that is called in case you can utilizing the fungal cells and all are the 14. That is the biogeological cycles. Biogeological cycles, these are the decompositions and decompositions are there, and that one. Another thing is the uh, another thing is that one. mm, Another one is called, that one is the another for natural would nature of the possible without that one, and without that one set. Here, the cycling of biogeological cycling of carbon in nature. Carbon in nature. Carbon in nature, and that one acting as a primary decomposers of the organic material. Furthermore, in agricultural operations of fungi play uh, important role as the mutualistic symbiotes. This is the pathogens and sporophytes, where they mobilize, nutrient and affect the physicochemical environment, and can be exploited, that is the nature of the environment, mobilize exploited, and against of biocontrol as a biofertilizers. As a biofertilizers, fungal metabolism is also responsible for the deoxification of the organic pollutants and for removing bioremediation, heavy metals, and other reclient chemicals. And this one, reclient chemicals, environment, the production of many econ- economically important industrial commodities reels on the exploration of yeast and fungal metabolism, and these includes such design products as whole food additives, fermented beverages, antibiotics, probiotics, pigments, pharmaceuticals, uh, biofuels, enzymes, vitamins, diseases, organic and fatty acids, and sterols. More natively fungi can considerable disease. spoilage and decay of important commodities and materials, building cross food supplies. In terms of human health, some yeast and fungi represent major opportunities. Life-threatening pathogens with list other other our life serves as they provide automatically antimicrobial and chemo chemotherapeutic agonists. In modern biotechnology, several yeast several yeast species are being exploited as host for the experience of human therapeutic proteins. Following recombinant DNA and gene editing technology. Recently, the application of gene editing using that is a CRISPR-Cas mechanism leading to revalence fungi genetic engineering. Further, an international synthetic biology research consortium called SC0.20 has embarked on the construction of the this is especially the case of now numerous fungal genomic and proteomics is providing valuable insights into the human genetics and heritable disorders. Knowledge of cell is many currently known fungal genes. This is called synthetic ones are fully elucidated. It is apparent that the fungi are important organism for human society, health or well-being, and studies of fungal physiology are very pertinent to our understanding, control, and explanation of this group of microorganisms. This chapter describes some basic aspect of fungal cell physiological, focusing primarily on nutrition, growth, and unicellular yeast and filamentous fungi. Morphology of yeast and fungi. There are a diversity of yeast and fungal cellular morphologies. Most higher fungi are filamentous. Yeast grow as unicells and some primitive fungi such as the Tritridomycota grow as individual rounded cells. Dichotomous branched chains of cells with root-like rhizoids for attachment to a nutrient resource. Here we consider the most common growth forms, the filamentous fungi and unicellular yeast. The filamentous, the cross morphologies of macrofungi and microfungi are varied and often apparent throughout the environment. For example, we can easily recognize a variety of mushrooms and toadstool. The sexual fruiting bodies of certain macrofungi. The higher fungi, Ascomycota and Basidiomycotin and related forms. During a walk through patch of woodland, microfungi, that is the mold, are also derived and often observed on decaying food and detritus, whereas many, including the colored rust, smut, and mildews, are common plant pathogens. Closer inspection of these visible structure, however, reveals that all are composed of aggregated long branches, threads, hyphae, organized to support spore for and dissemination. The hyphae of these aerial structures the supporting substratum as a network termed a from which the apically growing hyphae seek out, exfoliate and translocate available nutrients. Apically growing hyphae usually have a relatively constant diameter, ranging from 1 to 30 more depending on fungal growth conditions. Fungal mycelium most evidence extending from an inoculum on within and sometimes above the substrate, forming a near spherical three-dimensional colony. This radiant circle is also visible during the growth of fairy ring fungi in the Europic fungi, eukaryotic fungi, therefore, spatially, temporally, indeterminate organisms. Unlike animal, plant, and other micro individuals, have no me- the mycelium not, however, simply a homogeneously extending different different interconnected regions of the fungal mycelium may grow branch anastomose. That is a fuse, age, age, die, and sporulate, sporulate and display varying physiological and biochemical activities as different times or even simultaneously depending simultaneously depending on local micro environmental conditions. This called growing on relatively media may be pigmented, exhibit different morphological sectors, produce aerial structures, grow as the fast effuse are and the forming forming a channel running alone at the organism. Peripheral hyphae are often closely packed and melanized for filamentous fungi, and yeast are simply different styles of fungal growth suitable for occupation of different occupation of the different that is the different certain important different habitats and produced produced by habitat and produced by different cell growth polarity. Many species termed dimorphic fungi can adopted either the hyphal or unicellular yeast forms according to environment circumstances. For example, certain important human and animal pathogens exist as yeast forming mobilized in body fluid but are able to form hyphae, pseudo hyphae, tissue invasions. And what is the yeast? Yeast are unicellular, mostly Ascomycotina, Basidiomycotina are members of the Didyromycotina groups. Fungi that divided actually by budding for fusion and whose individuals cells can vary from very widely from 2 to 3 millimicrons to 20 to 20, 50 millimicrons in length, and 1 to 10 millimicrons is the width. The Saccharomyces cerevisiae, commonly referred as to brevier or baker yeast, is a generally ellipsoidal in shape with a large diameter of 5 to 10 millimicrons and small diameter or round 5 millimicrons. This is the great diversity in shell shaped and modes of a cellular reproduction in the yeast. As a summarize in the yeast, the morphological agar grown yeast, great diversity in terms of color, texture, and geometry of gained colonies. Several yeast are pigmented and the following colors. The colors may be visualized in surface grown colonies. Cream, white, Cercospora cerevisiae, cream color. White, Geotrichum candidum. Black, Eriobasidium pullulanus. Pink, Fascia rhizodozamine. Black, black color, Aerophysalum. These are the yellow color. All are the pigments of some yeast have biotechnological uses, including astaxanthin from Penicillium rhizodazamide in aquaculture feed supplement for the farmed salmon fish. Called salmon fish. Next one, the fungal surface. The cell envelope is yeast. The yeast and fungi is the peripheral structure that encase the cytoplasm and comprise the plasma membrane. The periplasm, cell wall, and additional extracellular structure components such as the fimbriae or capsule. The cell wall represent environmental and defend as a directional growth, cellular strength, shaped, and Neurospora carsa. For example, Neurospora carsa. The wall is thin and the effect become thicker. And the plasma membrane components of the fungal cell enveloped phospholipids bilayer interspread with globular proteins that dedicates entry of nutrient of exit of metabolism and represent a selective barrier for the translocation. Ergosterols is the major sterol found in the members of fungi. The most important of the fungal. Ergosterol is the, in the fungi, sterol's name is called ergosterol. In contrast to the cholesterol found in the membrane of animals and phytosterols in plants, ergosterols in fungi. This distinct is exploited during the use of certain antifungal agents used to treat some fungal infections and can be used as an assay tool qualifying fungal growth. The peripheral periplasmic space is the region external to plasma membrane, internal the cell wall, internal the cell wall in yeast cell, and it compresses created proteins that is called nanoproteins, enzymes. Enzymes such as the inverse acids, inverse acids and plasma membrane and is the region. The periplasm in the region, the east, the enzyme, the filament fungi is the maybe fungal seal surface topological features. That is the same. Nanometric resolution achieved using the automatic force microscopy.
This whole almost 75% that it's different story. It's amazing how by just changing the order of the same meal, it can increase the chances of losing fat by more than 50%. But how does this happen? I mean, after all, everything that we are eating is going into the stomach. But what scientists have found is that when we start our meal with a plate full of vegetables, the fiber in veggies coats your intestines, creating a sort of protective mesh, a kind of shield on the walls of the intestines. And that shield slows down the speed at which the rest of the meal will be absorbed in bloodstreams, and you will get constant energy for a longer period of time. This keeps you full for longer and prevents unhealthy food cravings. So a simple rule, if you want to melt that fat faster, eat whatever you are eating. Just start your meal with salad, a plate of carrots, cucumber, or whatever you feel like having. First eat the salad and then start eating roti, sabzi, rice, dal, whatever it is. Just a simple thing, and the speed at which your body reduces fat will increase by 75%.